All right, so the way we've decided to break up the time today, I'm going to begin by introducing the basic idea of chromatin and the basic idea of chromatin immunoprecipitation. And of course, a lot of the time we do chromatin immunoprecipitation as a prelude to making libraries for next-gen sequencing, the so-called process of ChIP-Seq. But I'm not going to talk at all about the analysis of the data we get from ChIP-Seq. I'm going to have maybe one slide that shows ChIP-Seq data, and then Noam will pick it up and talk about how to go from having a ChIP-Seq library to having analyzable ChIP-Seq data, and talk about the significance of ChIP-Seq, the kinds of things we discovered. So to begin, I'd like to begin historically with coined the word chromatin. It was coined by this man, Walter Fleming, not to be confused with the Scottish Fleming who discovered penicillin, working in Germany and in the late 19th century, where it became a lot of biological problems started to be tackled by putting stains and chemicals on cells. And he worked with aniline dyes and was able to generate pictures like this. And he's known as the father of genetics, and he associated chromosomes with chromosomes. He coined the word mitosis, but he actually did not associate chromosomes with heredity. Does anybody know who did associate chromosomes with heredity? Because I didn't put a slide in on that. I'm not sure, actually. I think it was started then working at Columbia with fruit flies in the 1920s, but I'm not totally certain. Okay. But the next point I want to make is that we have this idea about chromatin as a complex of DNA and protein that's in the nucleus of the cell. But it's interesting to me that it was not appreciated until the middle of the 20th century which of those two factors actually was responsible for heredity. So the discovery that chromosomes were involved in heredity was in the 1920s. The discovery that the DNA of the chromosome was responsible for heredity in the late 1940s. The experiments that were key to that are basically outlined here, going back to the earlier work demonstrating something called transformation, which is the transfer of a phenotype from one organism to another. But when that was originally demonstrated, it wasn't known whether the transforming principle was the DNA or the protein. And that was established with the work of Avery, McLeod, and McCarty in 1944, showed that if you protease treated these filled nuclei, they were still competent at transformation. But if you nuclease treated them, they were not competent at transformation. So, again, back to the word chromatin, colored stuff in the stained cell. You saw the picture. This is a HERT stained cell. HERT being a dye that circulates in minor group DNA. And what's interesting about the picture is you see something going on here. And of course, we recognize this as chromosome condensation associated with mitosis. And it's related to a point I want to make, which is that although we all think about gene expression all the time, chromatin actually has a very complicated life cycle and a lot of very, very important jobs to do. And just about everything everything that chromatin is involved in, we can study it some way or another using chromatin immunoprecipitation. So what are the jobs of chromatin? Obviously, we all think of it as a molecule that stores information. Double helix of DNA, the sequence of DNA is the information storage form of the cell, and then a code schematized in the middle. But it has other jobs too. Again, related to information storage, physical storage, compaction. How does all that DNA fit in the cell? And the answer is it has higher order structure so that it can be a little more dense and the space available. Another aspect of storage is the fidelity of storage. How does DNA stay relatively unmutated over time? This particular picture shows foci of H2AX, which is a variant histone protein. I'm getting ahead of this here. We haven't talked about this yet. But these are, repair of double-strand breaks, which in this experimental context are induced by gamma radiation. chromatin is involved in the expression of information. This is the thing that I would say half of biology is devoted to for the last half century, study of gene expression, and which is simply symbolized here by unwinding the double helix with the DNA polymerase, moving across it. Okay, thanks. The expression of information leads to the possibility of differentiation. The fact that not all cells in a metazome or organism are the same because they express different genes. Not all the stored information is used in the cell type. How is this achieved? Chromatin has a major role in how this is achieved. Chromatin is essentially a repressive structure. It's a structure that prevents the expression of genes except in places where it's permitted. And this picture shows, I think, just a beautiful example of differentiation in the case of metamorphosis or tadpole frog. Finally, there's the transmission of information through the process of replication and segregation, which is called mitosis. Here you see a typical image of segregating condensed chromosomes. And then, of course, there's the process of recombination, which is typically associated with meiosis and gametogenesis, which is photographed there. So chromatin is involved in all these processes. It has a very complex lifestyle. One of my teachers once said the study of biology is the study of technology, and what he meant by that was not spectrophotometers and DNA sequencing machines, but rather the machinery that makes all of these things. So, taking a step closer to what is chromatin, the key component, the key protein component of chromatin are histones. Histones are organized into octets of these four subunits, each to a DNA wound around. You see there are certain variant histone proteins that you can hear all about. H2AX already, H2AZ, which is a gene, very central region. And H3 comes from three actual components, 3.1, 3.2. And also mentioned central A, this is H3 variants of the central variant. This is a picture of the co-crystallization of DNA. This is going back to the trial of 1997. And you can see that the four color-coded histone subunits and structure of DNA blue and yellow blue. One of the interesting or important insights that came out of the crystal structure of DNA of this complex was the observance of the localization of these tails, the amino-terminal tails of histone proteins. It showed that these tails are relatively solidly closed. It gave some insight into why the post-translational modifications of these proteins are so important to the regulation of function of these. So the tails of histone are extensively modified post-translationally. This particular slide focuses on histone H3, where the modifiable residues are schematized here. Particularly arginines, threonines, and lysines, and serines. So the serines and threonines are phosphorylatable, and the arginines and the lysines are methylatable. Lysines are acetylatable. So all of these modifications take place and are responsible for regulating the actual function of chromatin, enabling it to perform the diverse functions that I summarized earlier. There's over 100 different isozymes of histone modifying enzymes known at this point. Deacetylases, demethylases, their occurrence, expression is tissue specific, it's developmentally regulated. These proteins are frequently amplified in cases of rearranged cancer, implying that the aberrance of cancerous cells is, in a small measure, a result of modification of the biology of chromatin, almost certainly the key aspect of that is the regulation of gene expression. And interestingly enough, a number of these enzymes are druggable, so that's becoming a focus in cancer biology for sure, as well as in other therapeutic discovery areas. So, I want to show this slide. This so-called histone code goes back to the work of David Alice. And this was published in the year 2000, summarizing a lot of thought in this area. And this is an idea that has fallen out of favor, but in spite of that, it's essentially correct in many respects. And that is that the modifications of the histone proteins have specific, recognizable, stable meanings. So, for example, the translation of histone H3 and lysine 4 is associated with the initiation of gene expression of transcription start sites, etc. The translation of histone H3 and lysine 36 is associated with transcription. These associations are recognized by these authors and are still true today. The idea following the favor is that the word code denotes a sort of one-to-one translation. And in fact, it's not the case that there's a one-to-one relationship between these modifications and the functional state of the chromosome. And interestingly enough, a number of us just returned from a meeting in Palo Alto where Charting the Epigenome showed an interesting slide in which she actually functionally tested using a cancer assay, scanning that cell, and she put every little bit of control region into an expression construct. You can see from that data that many modified loci, for focus, contact, which are not active, fewer than more. So the modifications were, I gotta say, they were not obligate predictors of functions.
So to summarize then, chromatin is not an inert structure, but rather an instructive DNA scaffold that can respond to external cues to regulate the many uses of DNA. Are we over here?
Sorry, do you want to use this?
I'd rather not if I don't have to. But,
Okay.
But I'll just try to project. I like to raise my voice instead of hearing it so booming. And a principal component of chromatin that plays a key role in this regulation is the modification of histones, which, as I said before, are the principal protein component of the chromatin. And there's an ever-growing list of the modifications, and the complexity of their action is only just beginning to be understood. Nevertheless, it's clear that histone modifications play fundamental roles in most biological processes that are involved in the manipulation and expression of DNA. And transcription is certainly the best characterized process, which is regulated by histone modification, but by no means the only one. So what is responsible for these modifications to histone? So this is a sign I'm indebted to Alon for. Alon and Oren, I believe, made this. And it illustrates the point that in this picture, these are octamers with tails that are modified with these modifications. And there are protein complexes that physically associate with these modifications, which these complexes contain enzymatic activities that are capable of both modifying these tails, the so-called writers of the modifications, but also by binding to these modifications, reading these modifications. And so in a slightly more detailed view of this, it comes from this figure from Bannister and Kouzarides, in which these different modifications, and this picture focuses on lysine residues with the exception of serine 10 here, and shows the names of the domains of proteins that interact with those modified residues. So the chromodomain proteins, for example, are involved in the binding of methylysine and so on. The bromodomain proteins involved in the binding of acetylated lysine and so on. So this is how the histone code, if you will, or the modifications of histone are translated into biological functions. I'm very interested in this figure from a paper by Jake Jaffe here at the BroadE, an article by Peach et al., which looks at, using proteomic methods, the population of modified histones in a cell, and makes the point that it's possible to measure the fraction of proteins that bear any particular modification. So, and I've just basically taken this one part of it and blown it up to make it easier to see. So on a cell-wide basis, if you look at lysine 4 of histone H3, the vast majority of it is unmodified, and then of the modified fraction, the most abundant modification is monomethylation, and then a small minority of it is di- and trimethylated, and then also a fairly small amount is acetylated. So this is important when you start to think about chromatin immunoprecipitation, because the ability to get a specific signal in a chromatin immunoprecipitation experiment is dependent on both the representation of the modification you're interested in the cell, but also it's dependent on the off-target effect of the antibody you're using. So if you're using an antibody to histone H3, hypothetically, which has a small propensity to interact with, with trimethylated lysine 4 histone H3, but which has a small propensity to interact with, hypothetically, with monomethylated form, then even if it's a small tendency to cross-react from the perspective of the affinity of the reagent, the fact that this other modification is more abundant can lead to a significant degree of a contamination in your signal. So these pictures are very useful to understand what the liabilities are of doing ChIP with antibodies in a world of the imperfect world we live in which antibodies are typically not completely specific to the intended modification. So transitioning now from talking about chromatin to talking about chromatin immunoprecipitation, this was the first paper that used chromatin immunoprecipitation going back to 1985. And it, as they say here, they describe a method for examining the in vivo distribution of a protein on specific eukaryotic DNA sequences. And they go on to describe this method in some detail and apply it using antibodies to DNA polymerase. And the method, as worked out here, was somewhat more long-winded than the method we use today. What we do today in 1 or 2 days, they took 5 days to do, but the method in broad outline is really the same as we do it today. Beginning with cross-linked cells, the recovery of nuclei, shearing of the DNA. We don't use a cesium chloride gradient anymore. And the immunoprecipitation step, and then the recovery of the DNA, RNase and proteinase treatment of the DNA, and then finally some kind of analytical method, which in 1985 was not next-gen sequencing, obviously. But so to simplify it somewhat, this is a very simple schematic picture of what goes on in chromatin immunoprecipitation. Again, starting with chromatin, schematized here. It's fragmented into little pieces. We strive to fragment them to pieces the size of a couple of histone octamers. Antibodies are introduced in order to pull down the complexes of interest. The DNA is recovered, and then it's subject to some kind of analysis. And the interest today is principally in sequencing as a form of analysis. So for the next few minutes, I'd like to go into a lot of detail from the perspective of a group that is doing a lot of high-throughput chromatin immunoprecipitation into all the methodological choices that we entertain as we pursue this method today. And I think that the value of this is for those of you who are using ChIP in your research and are contemplating using ChIP in your research and are confronted with a tremendous methodological diversity that you need to sort of wade your way through. What are some of the rationale between doing things one way or another way? So one thing you'll encounter is that there are two fundamental ways of doing ChIP with and without cross-linking, so-called X-ChIP for cross-linking and N-ChIP, which also stands for native ChIP. In the Bernstein lab, we routinely use X-ChIP. That's exclusively. But frankly, the majority of what we study with ChIP is histone modification. And when you're doing histone modification, both X-ChIP and N-ChIP have been successfully applied. If you look at the groups that do the most ChIP, you see a diversity of approaches used. KG Zhao using N-ChIP, Bing Ren using X-ChIP, the Bernstein lab using X-ChIP. But it's interesting to consider that N-ChIP may be superior in the case of certain epitopes that are sensitive to formaldehyde denaturation. And there are certain ChIPs that we've had a lot of trouble getting to work, but which others have shown it's because the epitope recognized by the antibody is denaturation sensitive. So I didn't learn that until I was preparing for this talk, and I'd actually like to go back and try some N-ChIP now in specific cases with certain antibodies. For transcription factor ChIP, only X-ChIP is really widely used, and that's because when people are doing ChIP with antibodies to transcription factors, they're very, very interested in the specific localization of the antibody on a specific DNA sequence that the transcription factor recognizes. So they want that high precision that's achieved by cross-linking the protein to the DNA. In contrast, when you're doing ChIP with antibodies to histone modifications, it's a somewhat lower resolution method. You're looking at a region of the chromosome and asking whether it's associated with that modification or not. And it's only possible to make specific inferences about the localization of these things based on amassing a tremendous amount of data. And finally, for chromatin-associated factors, which we also do ChIP for, we only routinely use X-ChIP as well, even though we're not typically discovering specific DNA sequences where they localize. Now, if you do use formaldehyde to cross-link, one of the things we've learned is it's important to use very fresh stocks of both the formaldehyde and the glycine that we use to quench the formaldehyde. Time and temperature are key variables. Cross-linking proceeds more quickly at higher temperature than it does at room temperature, but it may be possible to achieve equivalent cross-linking with longer times at room temperature, which is just a practical consideration if you have a way to do cross-linking in a fume hood with heating or not. But the fragmentation of the DNA proceeds very differently based on the extent of the cross-linking. And so we know from that there's a tremendous difference between DNA that is under cross-linked and over cross-linked in how it sonicates. UV has also been used for cross-linking. It was used in that original paper that I mentioned. Extraction of cells. There are methods in which you do a single-step complete lysis and basically, in one fell swoop, break open the plasma membrane and the nuclear membrane. Benefits of that are that it conserves material because you don't lose any material during wash steps. In contrast, a lot of what we do uses washed nuclei in which we eliminate the cytosolic proteins which might contribute to background. This is especially important if you're doing transcription factor ChIP of a factor that shuttles between the nucleus and the cytoplasm, because the cytosolic manifestation of that protein can tie up your antibody, and it would never lead to productive pull down of chromatin because it's the cytosolic form of the protein. So that's something to be aware of when you're doing transcription factor ChIP.
This precedes cross-linking, or?
No, the cross-linking is done while the cells are still alive, actually. It's as the way of killing the cells. But then once you have the cross-link material, you can then extract them initially with a detergent that merely lyses the plasma membrane and sediment out the nuclei, and then change the detergent milieu to lyse the nuclei as well. Elan, most typically working with very, very limited numbers of cells, typically uses a one-step method to avoid the loss of material with the washes. We're doing some of each as depending on the availability of cells. Although we're trying to converge on a single method that we can use for everything. So methods of fragmenting DNA. There's acoustic energy in some form. For a long time, people have favored Branson probe sonicators. We're in the process of transitioning to using the Cavaris instrument to sonicate our chromatin. We still have a diagenode batch sonicator, which some people still come into our lab and use. We found it very irreproducible. So that's a personal preference. Enzymatic fractionation has also been done using micrococcal nuclease, and this is typically used in the case of N-ChIP, but not in the case of X-ChIP.
Because it wouldn't work? I mean, why wouldn't micrococcal nuclease work with cross-linked DNA?
I'm not sure. Does anybody know? Okay. I mean, it's not that the enzyme is denatured because you add the enzyme later. But this shows what the DNA looks like after it's been sonicated. So it's not the thing that gets sonicated is chromatin, and then we recover the DNA from the chromatin in order to analyze its molecular weight size distribution. So these are traces on a machine called an Agilent bioanalyzer for anybody that might not be familiar with this. And so in these pictures, this peak on the right represents a high molecular weight fraction corresponding to this dark region on a virtual electrophoretic here. This is a capillary electrophoresis-based method, but it has a synthetic image that as if you would run a gel. And as you can see, as you go from 2 minutes to 4 minutes to 6 minutes sonication, this peak shifts from the right to the middle and a little bit farther to the left. And we try to optimize the quantity of material that falls between these two vertical green bars. But as you can see, you take a single type of cell and you cross-link it on 2 different days, and the optimal amount of sonication that is necessary to get the material you want might be different. And so for that reason, we repetitively do this process of optimization. We don't feel we can just assume that all chromatin will sonicate exactly the same.
Do you do any RNase digestion at all? Do you think it's crucial?
We do RNase digestion as at the end of during the recovery of the DNA. Oh, yes. In order to this assay here is an analytical assay. It's not a preparative assay. Okay, this is a way of seeing what you achieved when you did sonication. This is not part of the workflow of the ChIP experiment itself. Okay, so this is that clear? Okay.
So in your workflow, you usually do RNAs?
Yes, in the actual workflow, after you've done the pull down, I'll get to that in a minute. But as part of the elution step, there's the reverse crosslink by putting the material at 65 degrees for 5 hours to overnight, and during that time, it's in the presence of RNase and protease. Okay. Another choice that you have to confront when you're doing ChIP is what kind of antibody you're going to use, monoclonals or polyclonals. The vast majority of our work has been done with polyclonal antibodies. And the key advantage is that far more of the antibodies known to work are polyclonal antibodies. But the disadvantage of these is that there's lot-to-lot variability because of the way polyclonal antibodies are made, and the need to validate the antibody is therefore a repetitive task to know as you purchase new lots of antibody from some vendor that the lot has the specificity that you want it to have. In contrast, monoclonal antibodies are a very intriguing alternative that we're beginning to explore now because of the fact that you might have less lot-to-lot variability. But it's harder to find ones whose epitope is not impaired by the fixation. So it remains to be seen, I would say, at this point, how successful we're going to be at introducing more monoclonal antibodies into our process. Now, I mentioned the need to do validation. So here's one of Holly's slides showing our favorite method at the moment for how we do the validation of antibodies that recognize histone modifications. And that is using this array of modified peptides that we purchased from Active Motif. And so this is an example of a particularly nice assay in which an antibody that's sold as an antibody that recognizes H3K4 trimethyl is hybridized to this assay. And when the data are analyzed, we see that the only really significant cross-reactivity of this particular antibody is against histone H4 lysine 20 trimethyl, which is a very low abundance modification in real cells. These peptides are synthetic peptides. So this assay can show us that an antibody recognizes something that might not even occur in a cell. So we're very, very happy with the specificity of this particular antibody, because in practice, it won't recognize other things in real cells.
How many antibodies look like that are used for histone modifications?
Well, that's a particularly nice one. Howie, you might remember which exact one that was. I don't think I took notes. It's an Abcam antibody. Okay. We recently got our hands on a series of antibodies from Diagenode, the people that make the sonication technology that we don't like, but these antibodies appear to be very selective, or very specific to the intended modification. We are starting to use them, and we're starting to get some insight from the tracks that are coming back to us how good they really are. That's another way of validating antibodies, is once you learn to recognize the signs of a certain type of cross-reactivity in the actual ChIP-Seq data. But that's outside the scope of this talk, because I'm not talking about the data. Yes?
Sorry, was that fragmentation profile generated by what system?
I think I need to go back a slide here. You're asking me about this slide? Okay. This work done in 2008 was probably done using the Diagenode bioeruption system, just based on the date. Robin provided these slides to me, and I didn't have a chance to ask her that question. This was before I even worked here, so I don't really know.
So the covariates may be more consistent.
The covariates is also going to show. No, see, I don't. It's an interesting question you raise. The inconsistency between this and this is probably a function of the way the cross-linking was done, not necessarily a function of the way the sonication was done. So, you know, if your formaldehyde is a little more fresh, if the temperature in your incubator is a little higher, it can create variability in the extent of the cross-linking you achieve in a defined amount of time. And that will be manifest in the progress of how the sonication goes.
Yeah, a lot of our application, we really don't have enough material to do this optimization.
It's a real problem. And so Alon, who's a lot of his research, yeah. So the question was raised, if you're working with such rare cells that you can't waste the cells to optimize the sonication, what do you do? And the world's expert in that is sitting in the corner back there. And the answer is shoot for the middle and try to do things as reproducibly as possible. But that's a very real problem, which we've, frankly, you know, as recently as last week, we ran into a manifestation of that problem where we were trying to work with about 30,000 cells in a chip. And it's a tricky problem. So I think it was here. Just one of the things people talk about is what order should you add the antibody to the preparation? Should you start by complexing the antibody with the chromatin and then subsequently introduce the beads, or should you start by complexing the antibody with the beads and subsequently introduce the chromatin? And frankly, in most cases, they both work. But some people have argued that the antibody does a better job of diffusing to a counter-edge recognition site if it's not first coupled to the beads. And so that is the method that we try to use and what we're migrating toward using as much as possible. Recovery of DNA following ChIP. Here, there's not so much methodological variability. One of the changes in our protocol in the last couple of years was the transition from using phenol to using spry beads. So we've excluded phenol as much as we can from our preparation, just because nobody likes to work with it. But I see Casey sitting there, and I know she still gets some great results with phenol. So, Illumina library construction, I don't really have anything to say about this. It's very straightforward. We do it the way it's done at the platform here. And these are the steps, obviously. N repair, A base extension, adapter ligation. I'll comment that we do use multiplex adapters because it enables library pooling, and in modern next-gen sequencing, we would get too much data from a library if we were not able to multiplex our sequencing. And we most typically are making pools of 12 libraries and putting them onto 2 lanes of a high-seq, and therefore allocating it. We're about 1/6 of a lane per the sequencing of a library. And from this, we're getting on the order of 30 million aligned reads on average. One of the improvements that we'd like to make going forward is to get more reads of more distributed marks and fewer reads of less distributed marks, because really what we want to do is not have an equivalent number of reads for each immunoprecipitation, but rather to have an equivalent chance to discover the enriched regions that actually exist in a cell. And we know that we need more data to discover certain kinds of modifications than we do to discover other kinds of modifications based on the fraction of the genome that is tied up in that particular modification. So modifications that are occurring rarely or in a very punctate fashion, you can saturate your discovery of these things with fewer reads. So we just haven't figured out yet how to fine-tune the way we do pooling of libraries to get more reads of certain marks and fewer reads of other marks. But I think that's it would be a valuable step for us to take just in terms of the economy of the sequencing process.
Are you using the pay-RAND single length, and what's the solution?
Yeah, I think that's my next slide. Everything we've done up until recently has been with single-end sequences, 36 base pair. This is a typo. I made these slides very in a rush. We've been using 36 base pair single-end sequencing. Okay, but at this meeting we just attended, the annual meeting of the ENCODE project, Mike Snyder said, we should all transition to paired-end 101 base pair sequencing, so that in 2017, when we analyze our data, we'll have the best possible data. And that led to a kind of a discussion of cost-benefit. What are we going to discover by going to this more expensive technology that we're not going to discover with this cheaper technology? And the answer is, for the most part, not much. But in spite of that, we may transition some of our library sequencing in appropriate experimental designs to paired-end longer read sequencing. The hypothetical advantages of longer reads have to do with alignability as one factor. In cases where you're looking at DNA that has a repetitive character, a shorter read might not align, whereas a longer read might align. But the other advantage that was articulated by Anshul, whose last name Kundaje, is that by sequencing longer, you get a chance to interrogate the sequence some distance away from the actual end of the fragment that you're studying, and that might be in the region that where genetic polymorphism is most likely to be interesting. Okay, so a lot of what we're studying at this point are the modifications associated with promoters, for example, and the in a population of individuals in which there's genetic heterogeneity, we are interested in studying, in appropriate experimental designs, the effect of genetic heterogeneity on the function of regulatory elements, such as promoters and enhancers. But with short reads, we can't really interrogate the variability that might exist because we're looking at a fragment that is too short. We're sequencing too little of the fragment, and therefore not getting into the place where the genetic heterogeneity actually occurs. One of the obvious advantages of paired-end sequencing is that it gives you a real data-based knowledge of the fragment size distribution in your library, and that may be good to know. But it's not essential to operating this process. So particularly if you go back a few years where next-gen sequencing was more expensive than it is today, the only practical solution was something like a 36 base pair or even 25 base pair single-end sequencing. It's only more recently that we could entertain the possibility of using paired-end methods and longer runs. But again, the cost-benefit analysis of this has not really been done yet, and the concern is that the amount of additional information that we'll get out of this, raising the price of sequencing by a factor of 4 is just a couple %. So, yes?
Yeah, does that have any implications for locating the part of the DNA that's actually wrapping around the histone, or is that always identifiable?
In mammalian studies, with the depth sequencing depths that we do, we tend to not have very precise information about how the DNA is situated on the histone. In yeast studies, where the genome is significantly smaller, just a couple % of the size of the mammalian genome, the sequencing depths that you can achieve do enable detailed phasing of histones, where you can actually say with some precision from an affordable quantity of data, where is the histone sitting with respect to the DNA sequence. So I think I said I would show one slide in which I showed examples of ChIP-Seq data, but I'm going to leave it to Noam to go into this stuff in much more detail. And so this is just to give you a feeling from some fairly old work by Tarjei Mikkelsen, but I think he laid this all out very nicely in 2007. The transcriptional states are encoded in the genome, and so here is a pair of tracks actually that's associated with a transcribed gene. You see a peak of K4 trimethyl activity and an extensive region of K36 trimethyl activity, which is a signature of a transcribed gene. This could apply also to a non-coding RNA, which has the same epigenetic signature as a transcribed gene. Here's a signature of a silenced gene in which there's essentially no K4 trimethylation in this instance, but extensive amount of K27 trimethylation. And finally, certain histone post-translational modifications associated with heterochromatin, in particular K9 trimethylation and K20 trimethylation. So this is close to the end of my remarks, but I wanted to show just 3 variant techniques that you may have heard about and which fall under the umbrella of ChIP. And I think ChIP is our topic here, so I thought these would be fair to mention very briefly. This is a method called chip-on-chip, which in many ways is the precursor method of ChIP-Seq that we use today. It's fallen out of favor because the quantity of cellular material that is required to operate it is greater and because it gives much lower resolution information. But there are still some groups that use this method, and essentially in this method the big difference is how you analyze the chromatin immunoprecipitate once you obtain it. And so that's shown here, in which the DNA that's been pulled down is amplified and labeled, and then once it's made fluorescent, it's hybridized to a microarray. And so these methods took advantage of the existence of high-resolution microarrays, both Affymetrix arrays and arrays based on custom oligonucleotide synthesis. So this method is called chip-on-chip, in case you're wondering. Another very recently invented method, which is growing in popularity and is, I think, an important method for the future, is called exochip. And exochip is, once again, it's a form of chromatin immunoprecipitation, but it's used in particular with antibodies to transcription factors. And in this method, once you have pulled down the DNA, it's subject to digestion with a very particular enzyme called lambda exonuclease, which has the property of chewing the DNA from in a 5' to a 3' direction. So you see that this blue piece is missing here, and this red piece is missing here. Okay, and then the DNA is recovered from this, and it is the crosslinks are reversed, and then it's very important that it's also denatured, meaning that if there remains any additional residual annealing between these two strands, that the denaturation step eliminates that annealing. Okay, and by so doing, you recover the information about where's the actual 5' end of these sequences. Okay, so that when you then do a PCR primer extension, the end that you reach is the end that was left by the lambda exonuclease digestion. Okay, and that's followed by a more conventional adapter ligation and next-gen sequencing. So in this method, the ability to essentially map in great detail the point that the digestion ended at, which is the point where the enzyme ran into the factor, leads to these really remarkable high-resolution patterns of the discovery of the in vivo motifs that a transcription factor is bound to. So in this figure here, in which I guess I didn't capture the color scheme of this figure, but these different colors represent different bases, A, C, G, and T. The fact that these columns occur once the data are aligned is an indication that the transcription factor has a very homogeneous binding site. And then in this cluster analysis, a few different binding motifs are defined, which are similar to each other, but they're not identical. So there's 1, 2, 3, 4 principal binding motifs for this factor. And this experiment is actually done in mammalian cells. The earlier work that Frank Pugh and his colleagues did was in yeast, but they've actually demonstrated that this method can be made to work in mammalian cells as well. And this figure here shows a little bit about the spatial resolution of what you can discover using this method. So this gray mountain here is the spatial resolution of which you can discover using chip-on-chip methods, which I talked about a moment ago. This green peak here shows what we can discover with ChIP-Seq, and then this red spike in the middle shows what you can learn about the resolution of the binding site of an antibody using ExoChip. So again, in the ENCODE consortium, as it's moving forward, there's a tremendous interest in adopting the use of this method when appropriate for ChIP with transcription factors. And I think this is my last slide, just one more flavor of ChIP, and this is chromatin interaction analysis by paired end tag sequencing, which again is a form of ChIP. It starts with pulling down of cross-linked fragments using an antibody, but then taking advantage of the ability to ligate adapters to these ends that exist in this complex, and ultimately by sequencing across these ends with these known sequences that have been ligated to them, making discoveries about what are all the ends that exist in some close proximity in this original complex. So this is a method that is used to look at the proximity of specific chromatin fragments in a cross-linked chromatin complex. And it's given some insight into I suppose it would be called the quaternary structure of chromosomes in a living cell. And I think I'm out of water, so I'll just try to take your questions if you have any. If they ask you anything you don't know, just say it's due to epigenetics. Okay, well, thanks for your attention and your questions as we went along. And if you don't have any more questions now, I'm sure they'll come up later, so feel free to ask at any time.